Welcome back. I hope you enjoyed your short break. Now we're going to be focusing on a very important group of proteins called enzymes. Enzymes are molecules that speed up chemical reactions inside your cells. And we call them catalysts, but biological catalysts. All enzymes are proteins, and therefore they have the same characteristics as proteins. All proteins are going to be heat sensitive, for example, and pH sensitive. But it's very important that you remember that enzymes, like proteins, are made up of amino acids. Now, in living organisms, enzymes speed up the biological reaction, and how they do this is by lowering what we call the activation energy. Now, have a look at this little picture. The little frogs in this pond over here need to get into this pond. And in order to get from this pond up here into this pond, they have to jump over a pile of rocks. And it takes a great deal of energy to make that jump. What would happen if we lowered the rock barrier and we made that rock barrier much lower? Well, we'd find that it's far easier for the frogs to jump over the barrier because it doesn't take so much energy. And so the frogs would be able to move over the barrier much quicker. And this is precisely what an enzyme does. An enzyme knocks down the amount of activation energy that is needed for the reaction to take place. And so if we looked at a graph, not looking at frogs anymore, let's get science now and scientific. If we look at a graph that shows the amount of energy that it takes for a reaction to go from reactant to product, we see that we needed, for example, that much energy. That's the activation energy. With an enzyme, you can see the activation energy is lowered, and because we do not need as much energy to get that reaction going, it speeds up the reaction enormously. Some enzyme reactions in your body put things together. They take two separate molecules and they join them together. They bond them together in what we call a synthesis process. Synthesis means to put together. So here we have a graphic that shows us that we have two substrates. Those are the molecules before the reaction. They are put together with the enzyme and we get a product at the end. The two parts or the two pieces of substrate have been joined to make a product. Another kind of enzymatic reaction is a degradation process, and degradation is exactly the opposite of synthesis. Synthesis was putting together, degradation is breaking down or breaking apart. And here in a degradation process, we start with one whole substrate, it fits into the enzyme, and it leaves as two separate products. The big substrate has been broken down into two products. A term that is important that you learn is that the enzyme exists by itself. When it is locked together with the substrate, it is called the enzyme substrate complex. And when the products break free, we're left with the enzyme, which can go and perform the repeat of this reaction many, many, many thousands of times more before it wears out. Now, for many years, scientists wondered how that enzyme substrate complex was formed. It would be wonderful if we had a microscope that was so powerful that we could see the individual molecules, but we don't. So what they did was they proposed a hypothesis, and they said that the enzyme and the substrate, here's our substrate, let's write that there so that you remember it. There's the substrate and here's the enzyme. They said that the enzyme and the substrate fitted together like a lock and a key. That there was one key that could fit into the substrate the enzyme making the enzyme substrate complex, and that is actually how they fitted together. And we know now that that's not precisely true. It's not a case of a definite shape fitting into a definite shape, like Lego bricks. When you were a child and played with Lego, you knew, or doing a jigsaw puzzle, only one piece could fit into the other piece very precisely. It's slightly different. It's still similar to a lock and a key, but this theory is called the induced fit theory. And so we have the substrate and the enzyme, but when they fit together, the enzyme modifies itself and actually changes shape slightly to fit into or fit around the substrate a little more efficiently. So this is what we call the induced fit theory, where the substrate is one shape and the enzyme, as it locks onto the substrate, shifts in shape slightly to accommodate the substrate. And that's why I've shown it with this little graphic. We can see that that swimming costume looks nothing like the complex that it's going to fit onto. And that's exactly how the enzyme and the substrate do fit together, with a little bit of movement from the enzyme that holds the substrate in complex together. As the product is formed, so the enzyme relaxes and releases, and the product can move away from that enzyme substrate complex. Now, after the break, we're going to look at how proteins are made. And you're going to see that this is an extremely complex process. It's a process that, even though it's making proteins, relies on enzymes to actually control each step of the process itself. So after the break, tune in once more and we're going to look at protein synthesis.